So as I told people, I don't have myasthenia gravis, but I've had a thymectomy, I've had complications of steroids, I've had a lot of the problems that people with MG have, so I can sort of feel your pain because I have felt your pain. I just want to add a couple of points. I may look old because I am, but one of my heroes when I was in graduate school was John Lindstrom. John Lindstrom and Jim Patrick did a phenomenal experiment when I was a grad student, and they were looking to isolate acetylcholine. They wanted to make antibodies to the acetylcholine receptor in order to isolate that receptor. And if they isolated the receptor, maybe they get a Nobel Prize, maybe not. But anyway, what happened was that they found that the animals that were sensitized to make antibodies to acetylcholine receptors, and these were bunnies, developed a condition that looked like myasthenia. And that study, that serendipitous study, was what really nailed the idea that myasthenia for most people is an autoimmune disorder. And I was inspired by that, and it directed what I did henceforth. Gary Cutter he's a number cruncher. Now, that may seem kind of dull. You know, you think of accountants and you think of bookkeepers and that kind of thing. But really what people who do number crunching do is they help design a study so that it is structured in a way that it will yield useful information. If you don't have a quality number cruncher, the design of your study is going to stink. And you may put in an awful lot of work, but not come up with a conclusive answer. So I think that number crunching is incredibly important. Gil I've known for a long time, a Texas boy. I think he looks a bit like Clark Kent. The old TV Clark Kent, that's before his time. But the thymectomy trial was a phenomenal study. The idea of determining whether thymectomy was efficacious or not had been bouncing around for decades. Fred Jurecki championed that cause within the MGFA. John Newsom Davis, who Gil showed, was the initial leader and died tragically in the process of trying to get additional centers to participate. And this was truly an international study, and it also showed the ability of the MGFA to have an influence on research. The MGFA did not fund the large study. We don't have enough money to do that. But what it did is it funded the kindling. It funded the work that got the project off the ground. And believe me, it took a lot of work. There were a lot of submissions and back and forth with the NIH. And I think if the MGFA had not supported it at the beginning, the study might not have taken off. And it's also to Gil's credit, to the credit of all the people who participated in this, that it finished with a meaningful answer. A lot of studies that are funded never finish because they have trouble recruiting patients. And so it was fortunate that enough people were willing to participate in the study that the study came to fruition. And Dr. Nowak is a legend in his own time. I mean, he is somebody who is greatly admired by the MG community. One of my good friends, Sue Klinger, who's in New York, was a big advocate for Dr. Nowak. And if you know the New York area, people in New York, there's little communication between the boroughs. I mean, people in Brooklyn often don't talk to people in Manhattan. So for somebody in New York City to know about a person at Yale is amazing, and it's a reflection of how qualified he is. His patients love him, as far as I can tell, and whenever you need help, you can always depend on him to pull through. So this is a phenomenal panel, excluding myself. I mean, I'm just up here for show. But do you want to talk some more about the thymectomy trial?
I'd be happy to answer questions, but before that, well, thank you. But Dr. Ruff, for years, has done more to communicate what's going on the scientific side of research in myasthenia gravis to the general population, to all of you. In newsletter after newsletter, he's done that for a very long time. So he needs to get credit for that. And
okay, so as you so graciously heard a few minutes ago from Robert Ruff, once upon a time, long, long ago and far, far away, meaning 1973 in San Diego, we found that when we immunized rabbits with electric organ acetylcholine receptors, they got weak and fatigable. Methanon repaired them, looked very much like MG. Then we went on to use the same antibody assay that we used in those rabbits to find autoantibodies to acetylcholine receptors in MG patients, and that became the diagnostic assay. Since then, we've studied experimental autoimmune myasthenia gravis, EAMG, in rats. And with von der Leyen and Andy Engel and others, we showed that the pathological mechanisms that impair signaling between nerve and muscle and produce the weakness by destroying acetylcholine receptors in the architecture of the synapse is the same in EAMG and MG. And Socrates Tsartos and I determined the specificities of autoantibodies to acetylcholine receptors in EAMG and MG. And as you heard this morning, this resulted in discovering a main immunogenic region at the extracellular tips of alpha subunits. And this highly immunogenic region is pathologically significant because it's on the extracellular surface where antibodies can easily get to it to cause their destruction. And all of these experiments show that the ideal therapy for MG would be to specifically suppress the pathological autoimmune response to the MIR on the outside of the receptor, because now, as you've heard this morning, non-specific immunosuppressive therapy is used with prednisone. And that has the problem that it suppresses all immune responses. And not only does it take out both autoimmune and immunoprotective responses, it has all sorts of side effects, which we've heard about in some detail this morning. So what Jay Luo and I have been up to in the last few years is developing a specific immunosuppressive therapy that works in EAMG. And this therapy involves immunizing rats with the cytoplasmic domain, the part inside the cell, from human acetylcholine receptors. And so this therapy should be directly applicable to MG because we're already using the human antigen to do it. And we'd really love to be able to continue our studies and test this therapy on MG. So this slide shows you what you've already heard about today, that antibodies bound to the outside of these receptors can fix complement and cause destruction. They can cross-link the receptors and otherwise cause their destruction. And the complement-mediated destruction disrupts the postsynaptic membrane. And in the animal model, we can interrupt the synthesis of these autoantibodies that block the receptor And we're doing this by vaccinating them with the interior part of human muscle acetylcholine receptors. And we think this is safe because we can repeatedly immunize the animals with the cytoplasmic domains, and they don't get sick because they can make all the antibodies they want to that, and the antibodies can't get in the cell to bind to it. And the therapeutic immune response reduces the amount of pathological antibodies to the outside of the receptors by inhibiting, destroying the cells that make it. So we're not competing for them. We're fundamentally getting at the heart of the mechanism that produces the autoimmune response. And this requires using an adjuvant. Any vaccination you have uses an adjuvant to make the immune response stronger. And we have been doing all these animal experiments in incomplete Freund's adjuvant, which is a relatively mild adjuvant. It used to be used in flu immunizations in humans, but is not now. And the last slide I'll show you shows you that we can now use an adjuvant that's commonly used in human vaccines, which I think is the last real barrier to being able to think about doing this in patients. So in these data slides I'll show you, we start out on day 0 immunizing the rats with acetylcholine receptor purified from electric rays. And we do it in a very strong adjuvant titer max. And then at later times, starting a couple weeks later, in this case, we immunize the animals once a week with a therapeutic antigen. Now, after this initial immunization with electric organ receptor, there's a transient acute phase of weakness. And the specific mechanisms there differ from what happens in chronic EAMG or MG. But starting about 2 weeks later, untreated animals will get very weak until they're essentially at death's door. But if we have treated them, they're completely protected from the development of EAMG as shown in the orange data points. Okay, and the successfully treated rats are resistant to a relapse of EAMG. So if we wait 6 months and immunize them again in the very strong titer max adjuvant that we use when we immunize them with torpedo acetylcholine receptor, control rats again develop a very severe EAMG, but the previously treated rats do not. And it turns out the reason they do not is that although they make some antibodies to acetylcholine receptors, the isotypes of these antibodies are different. They don't fix complement. And that's why they don't get EAMG, because they don't lose receptors and the postsynaptic membrane isn't torn up. And so you can see here, these animals are very well protected and for a very long time. So we'd like to think that a short course of immunization might provide very long-lasting immunization, just as happens when you're immunized for anything. And of course, I showed you in the first slides where we started treating before the animals were really very sick. So the normal case is that folks come in and they're already quite weak. And that's what we've modeled here. So the animals were immunized and allowed to develop quite severe EAMG, and then treated with the therapeutic immunization. In this case, just 3 shots at 35, 49, and 63 days. And very quickly, the synthesis of antibodies to the main immunogenic region in the extracellular domain was shut down. This is much faster than you can achieve with most immunosuppressive agents. And basically sort of stopped the response in its tracks. And you can see that the net effect of this sort of therapy preventing development of the chronic EAMG has a huge effect on the rats. These are young rats who should be growing and gaining weight, and the treated rats that are prevented from developing EAMG gain weight and get large and strong, whereas the untreated rats are so weak they have a hard time drinking and eating, and you can spot the difference here quite clearly. Now, then we decided we'd push this as far as we could. So we immunized a lot of rats with torpedo acetylcholine receptor, and then waited a very long time, out to 92 days before doing anything. Okay, so this animal model is a very severe EAMG model, and by that time, half of those rats were dead. And then we split them up into two groups and treated one of the two groups, and again, that's in the orange. And so we start treatment here, and even after this many months of very severe response, enough to kill half of them, very quickly, the production of antibodies to the extracellular surface, the MIR, is suppressed, and they get much better. And all the data I've shown you to this point are published already. You see in our papers. This is an unpublished experiment. It was done quite recently. And here we've used alum. This is an adjuvant that's been used in probably most of the immunizations you've ever had, and it can be used in humans. And what we find here is it works every bit as well as the incomplete Freund's adjuvant in the experiments that I showed you before. So we wait till after 2 weeks, and the untreated animals go on to develop EAMG, and whether we start the treatment then in IFA or in alum, the response is greatly suppressed. So what we'd ideally like to do before treating humans is determine the therapeutic mechanisms in some detail, but that's not absolutely necessary. But we do need to try to optimize the doses and separation between the doses, and we might be able to alternate between giving doses in adjuvant and just the therapeutic antigen, because we think the therapy works in two ways. One, directly through the antibody, where immune complexes formed on the cells that produce the autoantibodies bind acetylcholine receptors shed from the muscle and are heavily labeled with the therapeutic antibodies, and that immune complex causes apoptosis. It causes that sort of antibody-producing cell to die. The other thing that can happen is the antigen produces regulatory T cells that suppress them, and probably both things happen. Another thing which we would do ideally if we could is test it on real MG, and real MG occurs in cats and dogs. One of my former postdocs is now on the faculty at UCSD and is sort of the center for doing MG diagnosis for cats, dogs, and tigers using the same assay that we use for humans, except using antigen from cats, dogs, or tigers. And cats are the ideal. Dogs have a spontaneous remission rate that makes doing these experiments very difficult. Cats, like humans, don't spontaneously remit. That's especially bad news for these cats, because then 58% of domestic cats that are diagnosed with MG are euthanized on diagnosis because people don't want to deal with it. But that gives a big baseline to improve the life of cats. But that would get over cynics who were, oh, you got an animal model. If we could make it work on the real thing, that would be nice. Okay, our myasthenic crisis is a financial crisis. And we've been having a hard time getting support. And if we don't get substantial support within the next couple of months, the university will close our lab, and none of us will find out the answers to what happens here. And so this shows our website. If you go to this website, you can get a description of what we're up to and references to all of the papers describing this work. And you or your rich friends can actually donate money here should anyone have the urge. So thank you very much.
1831. I was described as a number cruncher, and I didn't want to disappoint you. Now it's 1856. 1831 is when I put the slides together, which are in your book. That's how many patients and participants have signed up for the registry. The MG registry is really your registry. And I'm just, part of the reason to be here is to thank you. And the other part of the reason is to give you a little bit about what we're doing. I'm not going to go over the slides. They're in the book. You can look at them. They tell us a little bit about why we want to do it. What I want to say a little bit about why is a registry necessary? You know, there are very good clinical databases that have been collected in particular offices around the country. The problem with that is, how many of you go to Duke? They have a great registry, a clinical database. I don't see a lot of hands. That's part of the issue. Who goes where? So when you have a registry that's located at a clinic, we don't know whether that pertains to all patients or only the kinds of patients who made it to that source of care. So everybody's heard a lot about, we have an automated medical record. Clinicians spend a tremendous amount of time doing great data entry. And so why can't we use that data? Well, we might be able to do that someday, but currently those records are mostly for billing purposes, and I have the same problem trying to get that data from 5 different hospitals, because even though they might have the same medical record system, they don't talk to each other. So it's difficult to get that information out. And what happens if somebody goes to a different hospital? So you have, their records are truncated. So there's information there, and it will be useful. In the meantime, though, why don't we use insurance records? Insurance companies have tremendous amounts of data that they actually let people, and they have companies that do analyses to understand what's happening. Well, that's great. They have millions and millions of lives tracked over time. It has a couple of problems. One is it knows how much was charged. It knows how much they paid. It knows what they're paying for, but it has no severity, so it doesn't have any information about the severity of the condition that they're paying for. They don't also know about the outcome. And people may not keep the same insurance company, so your record, again, may get split from 1 to 1. So in the United States, we actually don't have record systems that allow us to track and report on the disease that occurs over time. European systems, many other systems, have centralized healthcare systems that allow them to follow patients over time, as long as you stay within that country. I mean, everybody has their own problem in terms of how to deal with this. But registries in the US can provide a constant source of information where the same patients from all different kinds of care providers can be looked at both cross-sectionally when you register and over time. And that's what we've started. We've actually, the NARCOMS registry was mentioned earlier, which was started in the mid-90s. And it's taken years to grow to where it is and get many of the things out. The executive committee and people around have used it. There are hundreds of publications that are in the scientific literature that people can benefit from that have taken place. But it didn't start doing that for almost 10 years after its existence, because it took that long to get everything together. We've been in existence about 2 years, realistically. And the data are being mined. There are 2 abstracts, one for the ANEM and one at the New York Academy of Sciences meetings, where we're looking at some of the questions to follow up the thymectomy trial to see, do we see the same results in the registry? We've had a number of companies, and why are companies important? Well, that's how we get new medicines. We do have the developments in the basic science that start here, but somebody has to carry it forward, because laboratories just don't have the resources to move them forward. So the pharmaceutical industry often wants to have registries available so that they can quickly get answers to some questions they have. They're not exact answers, but they find out how many refractory patients might there be. How many patients have a change in their quality of life over some period of time? And registries can provide those answers. And they're relatively quick. And while it's bothersome to all you, and that's where the thanks comes, you guys fill out these questionnaires every 6 months. You know, the programs don't always work right, and the questions sometimes are, strange. Why are we asking that? But the value of that data for the MG community can't really be measured. If we ever have a unified healthcare system that collects all this information and includes patient-reported outcomes and the kinds of things we collect, will be irrelevant. But I think all of us have a job for a long time if what happened last week is any sign of it.
What I wanted to do is discuss and present some of what we're doing in terms of the rituximab and myasthenia gravis clinical trial, also known as BEAT-MG. So I just have a few disclosures in terms of grant and research support for this study. The study is supported specifically by NINDS. We did initially get some seed funding from the MGFA early on, and I wanted to disclose relevant to this presentation that study drug and placebo have been given to us by Genentech through an investigator-sponsored trial agreement, but they have no role in the study design, implementation, or data analysis. So what's the rationale for looking at new therapies? So you've heard throughout the course of the conference that there's various therapeutics out there, including steroids as the gold standard typically, and things like pyridostigmine and mestinon and Imuran and Cellcept, et cetera. But approximately 10 to 15% of patients with myasthenia gravis are considered to be refractory. And that's sort of a moving target in terms of what defines individuals with refractory disease. The other rationale for looking at new therapeutics in MG is that there's a paucity of proven steroid-sparing agents. We saw the results from the MGTX study, which are extremely positive and encouraging, but in terms of looking at other immunosuppressive therapies, a lot of the studies that have been completed or closed early because of poor recruitment, in the end didn't really show a statistically significant difference between the two groups. And also a need for safe, well-tolerated agents and strategies that are targeted. So specifically looking at what the immune disruption is that occurs or is causal in myasthenia gravis and looking at that and targeting that. And the possibility of inducing tolerance. So the whole approach of vaccination and other therapeutic strategies, can we really effectively at the end of the day cure myasthenia gravis? So it's really how do we induce tolerance? Is that something that's going to be done this year or in the next 5 years? The answer is no, but is it something that we should strive and try to do over the next decade or so? And the answer is yes, and I think that the future is extremely encouraging. So what's the rationale? So B cells or autoreactive B cells are implicated in the pathogenesis of myasthenia gravis as well as other autoimmune conditions. So it makes a lot of sense that we should study a therapy that's targeting those B cells. There's been a lot of studies that have been looking at other conditions such as rheumatoid arthritis, lupus, multiple sclerosis, using medications, specifically rituximab, or those that are targeting B cells, with promising results. So the first report of rituximab's benefit in myasthenia gravis was essentially in the early to mid-2000s in an individual that also had lymphoma, and as such was treated with rituximab for their lymphoma, and it was observed that their myasthenia gravis also improved. So since that time, there's been a lot of excitement, a lot of case reports and small series that have looked at rituximab's benefit in the treatment of patients with myasthenia gravis. The most important thing is that, similar to the thymectomy study, a lot of these studies are small, with a few patients, and there's no placebo-controlled or control arm that we can actually say that there's a significant benefit as compared to individuals that did not receive that therapy. One of the earlier reports which came out of our group, we looked at 14 patients, 6 with AChR positive myasthenia and 8 which were MuSK positive. And we showed that those individuals had a significant improvement, both clinically as well as in their ability to reduce chronic steroid use. And so based on these preliminary data or pilot data, you can refer to them as, this was really what we used to design and implement or obtain funding for our phase 2 clinical trial. So what's our objective for BeatMG? The specific objective is to study whether or not rituximab is, number one, safe and beneficial in the treatment of MG, and whether or not it warrants a larger phase 3 trial, which is really going to more carefully explore its efficacy. So our target was to enroll a total of 50 individuals with generalized AChR positive myasthenia gravis. We targeted the AChR positive group because we didn't feel that we could successfully enroll a significant number of MuSK positive patients. The original study that we had designed before it went to NIH for review actually did include both groups of patients, but because of feasibility of enrolling or recruiting a sufficient number of MuSK positive patients, that part of the study was put on hold temporarily. So I don't want to get into too much detail in terms of the eligibility, but basically individuals of any age with generalized AChR positive MG, those individuals that are either on prednisone only or on prednisone plus another immunosuppressive therapy such as Cellcept or Imuran. So this is a placebo-controlled prospective trial with a 1-to-1 randomization. What that means is that 50% of the patients enrolled get assigned to the placebo arm and 50% get assigned to the rituximab treatment arm in this case. The study in its duration is 1 year or 52 weeks with 2 rituximab cycles, one initially after randomization and the other approximately 6 months later. So getting into more of the specifics, I already talked about the safety, but our primary outcome in addition to safety is steroid-sparing effect. So essentially looking at whether or not individuals at the end of the study or at the last 4 weeks of the study have a significant reduction in their prednisone as compared to the 4 weeks prior to entering the study or being treated with rituximab or placebo. And so we do know that there is a typical delay to benefit based on our observations with rituximab treatment, and usually that benefit is not observed immediately after treatment, but some weeks or even some months after treatment is initiated. So there's other secondary outcomes, things that you've already heard about in terms of the quantitative myasthenia gravis or myasthenia gravis composite score, and certainly other exploratory clinical outcomes. A lot of these were put in place in terms of our study to look at whether or not there is any signal or any significant benefit observed in any of these other measures to help us potentially design a larger phase 3 clinical trial. And certainly other things that we looked at, things like effects of chronic steroid use, so looking at BMI or body mass index, looking at an individual's hemoglobin A1C, at the start of the study as compared to the end of the study. There is one key component here that I don't think many people know about, is that during the course of our trial, we're looking at biomarkers and deep mechanistic studies to try to really understand how rituximab is effective, if it is effective, in patients with this condition. And I also try to tease out if there are individuals that are more or less or non-responsive during the course of our study. What were the distinct immunologic changes that were associated with those? Again, trying to improve our use of rituximab in the future. Which patient should be treated with it, which patient is less likely to benefit from it, to help us guide those types of decisions. So this is a very preliminary effort in addressing that question, but that effort is part of the BeatMG study. So this study, as I mentioned before, is supported by funding through the NIH. It's done through the NeuroNext Clinical Trials Network, which is basically 25 clinical trial sites that were pre-selected by the NINDS to run neurologic clinical trials. The beauty of this clinical trials network is, number one, the time to getting a trial up and running and implemented and getting patients enrolled in a trial is much quicker. There's master trial agreements already in place. There's a centralized IRB. So a lot of these things that delay getting a clinical trial up and running are already done in place. So this is a network that is used to doing other neurologic trials, and we were able to run our study through this trial. There were a number of participating sites. Basically, every single site that was interested in participating that was part of the NeuroNext Clinical Trials Network was allowed to participate at the end. So this is a brief overview of the study timeline. So initially, the funding was awarded to us in September of 2013. We initially activated a total of 15 clinical study sites. And by April 2014, we had our investigator meeting. We had all the sites basically up and running soon after that time. And our first site was activated in May of 2014. There was a delay in terms of when we initially got up and running versus when we actually got our first subject enrolled, and that delay was a number of months. So our first subject enrolled in August of 2014. This is not unexpected to me, and I was certainly expecting that there was going to be a delay, but this caused a lot of anxiety, I think, by many individuals, including the NIH, but we did quite well, and our enrollment target was achieved in April of 2016, so approximately 24 months after study sites were open for enrollment. Our projected was 24 months, and we were able to achieve that within the time that we had projected. So where are we? So I mentioned that we've already completed enrollment. So our 50th subject was enrolled in April of 2016. We technically enrolled 2 more subjects after that. So we have 52 subjects enrolled, with the last subject enrolling July of 2016, with our projection currently that last subject will complete their last clinical study visit or their week 52 visit in July of 2017. A little bit more on how we did in terms of our enrollment. So similar to the MGTX study, we looked at a lot of patients. So there were over 4,000 charts reviewed, over 1,800 subjects or patients specifically that were pre-screened. So these are individuals that were identified as potentially eligible with antibody-positive disease and on prednisone. And out of those, out of 1,800 essentially, we had 68 individuals that came in for a screening visit with signed consent, and of those 68, a total of 52 were randomized. So our recruitment rate was approximately 2 to 2.5 patients per month. This is how we did in terms of our overall consent. The dotted line is what we had projected. The red line is how we actually did. So early on, those first few months, as I said, there was a lot of anxiety about our ability to successfully enroll, but with the support of the MG community and certainly the MGFA, helped raise awareness for the study through social media, through blast emails, and certainly through some of the in-print MGFA newsletters. So this was certainly critically important. We tapped into other ways to raise awareness, but without the support of the MGFA and the community, I don't think this would have been possible. So thank you for all of that support. This is our randomization, which basically mirrors the consents that were signed for this study. So this is where we are as of late February. So of the 52 individuals, 38 have completed the study to date. There were 5 individuals that withdrew early for essentially reasons not related to the study at all or due to safety. And we have 9 patients that are currently ongoing, so they're within progress, so they haven't completed their week 52 or final study visits as yet. We have an extended follow-up study as well, which is looking at how individuals do after the week 52 visit. So if there's a significant improvement in terms of how they're doing clinically as well as a reduction in their steroid dose, well, where are they at 18 months and where are they at 24 months? So basically a year after completing the study. So this is critical information. We're trying to also understand how the B cells that we target and deplete with utilization of rituximab, what happens to those B cells as they come back. They typically start to come back roughly a year after the treatment is completed. So this is what everyone asks me about. Well, when are we going to have the results? So currently, I'm projecting that we'll have the results by late 2017. I have December 2017, but fully expect that sometime in the late fall of this year that we'll have those results available for public announcement.